Biologists at the U.S. Department of Energy's Brookhaven National Laboratory and their collaborators have discovered an aberrant protein that's deadly to bacteria. In a paper just published in the journal PLOS ONE, the scientists describe how this erroneously built protein mimics the action of aminoglycosides, a class of antibiotics. The newly discovered protein could serve as a model to help scientists unravel details of those drugs' lethal effects on bacteria, and potentially point the way to future antibiotics. Identifying new targets in bacteria and alternative strategies to control bacterial growth is going to become increasingly important, said Brookhaven biologist Paul Freimuth, who led the research. Bacteria have been developing resistance to many commonly used drugs, and many scientists and doctors have been concerned about the potential for large-scale outbreaks triggered by these antibiotic-resistant bacteria, he explained. What we've discovered is a long way from becoming a drug, but the first step is to understand the mechanism, Freimuth said. That gives us a way to study the mechanism that kills the bacterial cells. Then maybe a new family of inhibitors could be developed to do the same thing. Following an interesting branch, the Brookhaven scientists, who normally focus on energy-related research, weren't thinking about human health when they began this project. They were using E. coli bacteria to study genes involved in building plant cell walls. That research could help scientists learn how to convert plant matter, biomass, into biofuels more efficiently. But when they turned on expression of one particular plant gene, enabling the bacteria to make the protein, the cells stopped growing immediately. This protein had an acutely toxic effect on the cells. Interns could run experiments and see the effects within a single day, he said. And maybe they could help figure out why a plant protein would cause such dramatic damage. It was a strand of amino acids, the building blocks of proteins, that made no sense. That resulted in putting the wrong amino acids in place. It would be like reading a sentence starting at the middle of each word and joining it to the first half of the next word to produce a string of gibberish, Freimuth said. The result? All the bacteria's ribosomes make gibberish proteins. If a bacterial cell has 50,000 ribosomes, each one churning out a different aberrant protein, does the toxic effect result from one specific aberrant protein or from a combination of many? Breakdown of quality control systems could make aberrant proteins accumulate, causing disease. Messed up quality control. The next step was to find out if the aberrant plant protein could activate the bacterial cell's quality control system, or somehow block that system from working. They also discovered that the difference between cell life and death was dependent on the rate at which the aberrant protein was produced. When cells contained many copies of the gene coding for the aberrant plant protein, the quality control machinery detected the protein but was unable to fully degrade it, Freimuth said. When we reduced the number of gene copies, however, the quality control machinery was able to eliminate the toxic protein and the cells survived. The same thing happens, he noted, in cells treated with sublethal doses of aminoglycoside antibiotics. Model for mechanism. These experiments indicated that the single aberrant plant protein killed cells by the same mechanism as the complex mixture of aberrant proteins induced by aminoglycoside antibiotics. But the precise mechanism of cell death is still a mystery. The good news is that now we have a single protein, with a known amino acid sequence, that we can use as a model to explore that mechanism, Freimuth said. Scientists know that cells treated with the antibiotics become leaky, allowing things like salts to seep in at toxic levels. One hypothesis is that the misfolded proteins might form new channels in cellular membranes, or alternatively jam open the gates of existing channels, allowing diffusion of salts and other toxic substances across the cell membrane. A next step would be to determine structures of our protein in complex with membrane channels, to investigate how the protein might inhibit normal channel function, Freimuth said. This work was supported by a Laboratory-Directed Research and Development Award from Brookhaven Lab and in part by the DOE Office of Science, Office of Workforce Development for Teachers and Scientists, WDTS, under the Visiting Faculty Program, VFP. Retrieved April 29, 2022 from ift.tt/9-zarc4 DOE, Brookhaven National Laboratory. New model for antibacterial mechanism. Science Daily. ift.tt/9-zarc4. Accessed April 29, 2022. Thanks for watching. Please subscribe my channel for more videos.